Got it. Well, I guess we're ready to get started. Welcome to CureSHANK's first webinar of 2023. We're glad to have you here. I'm Geraldine Bliss, president and co-founder of CureSHANK. And most importantly, I'm mom to Charles, who is 24 years old and has Phelan-McDermid syndrome caused by the partial loss of the SHANK3 gene. This year, one of CureSHANK's goals is to help grow a community of people who share a common passion for improving the lives of people with shankopathies. And we're going to tackle this goal through several approaches. Our largest initiative to support this goal is our pre-competitive outcome measures and biomarker consortium. If you haven't already been in communication with us about it, please drop us a message and we will share all the details about it with you. Our consortium project manager, Trish Davidson, also on the webinar, will tell you more about the consortium at the end of today's webinar. Second, we want to enable collaborative and productive relationships among all of the stakeholders in our field. There are some really fantastic resources that have been funded by the NIH and developed by academic investigators. And our hope is that making those connections and introducing you to those resources will enable all of us to work with just great speed and efficiency to develop treatments for shankopathies. In our conversations with many different companies, we frequently receive questions about resources for iPSCs. Today, we are pleased to have two speakers who will share more about the resources that are available to you and the work that's already been done. I'm honored to welcome our first speaker, Dr. David Panchision, Chief of the Developmental Neurobiology Program at the National Institute of Mental Health. Dr. Panchision directs the funding portfolio in the developmental neurobiology and coordinates initiatives for the use of stem cells in biological discovery and therapeutics development at NIMH. I first met Dr. Panchision through a project with Dr. Joachim Hallmeyer at Stanford, where we collected blood and skin punch biopsies from patients with Phelan-McDermid syndrome. Today, Dr. Panchision will tell us about the NIMH stem cell program and the resources that are available to the scientific community. And so we're ready for you, Dr. Panchision.
Great, thank you very much. Let me go ahead and share my screen. And hopefully this is showing in presentation mode.
It is, it looks great.
Great, thank you. Great, thanks very much for the kind invitation and introduction. What I'd like to do is give a brief overview of what our repository does, then talk about specifically the Phelan-McDermid collection in the repository, and then give some information on how you access the data and biospecimens. So the NIMH repository and genomics resource was established in the 1990s. It plays a key role in facilitating psychiatric genetic research by providing a collection of well-characterized, high-quality patient and control samples from a wide range of psychiatric illnesses. This slide gives you an indication of what types of biospecimens are currently available and in what form. And this ranges from whole blood, saliva, and externally produced derived DNA, as well as RNA and cell lines in various types, including lymphoblastoid cell lines, which make up the largest proportion of the cell lines. We've introduced iPS cell lines in a little over the last decade, which I'll go into in a little bit more detail. In the bottom of this slide, you see that these are released in the different collections, typically based on the disease classification, but in some cases they're based on specific cell type formats like induced pluripotent stem cells. I'll also note at the bottom here highlighted that we have a very large collection of cryopreserved lymphocytes. These are not for distribution in the typical sense, but I'll go into a little bit more detail later about where that comes into play. So the NIMH repository is currently housed in New Brunswick at the Rutgers University Cell and DNA Repository. They took over the storage of the lines and the biomaterials in 1998. They've been transitioning to a company called Sampled over the last couple of years, and this is a company spinoff of the Rutgers University Cell and DNA Repository called Infinity Biologics, which went into a partnership with Roylance, and they're doing business as a company called Sample. And their facilities are moving from the Rutgers University campus to a facility nearby. The PIs currently, this is a cooperative agreement with NIMH. The PIs currently are Linda Brussowitz and Jay Tishfield. This has a data sciences component. As I indicated, the bioresources are stored with Sampled and processed with Sampled. And there is a clinical curation component that's led by both Linda Brussowitz and Veronica Wieland. I'll also note that over the last few years, the clinical and genomic data submission has shifted over from the NIMH repository to the NIMH data archives, although there's still going to be a significant component of the data still stored at NRGR. Just a little bit of history about how the iPSC component came into the NIMH repository. So as I indicated, the repository itself dates back from the 90s, primarily to enable genomic studies. But when human iPS cell lines were first reported in 2008, NIMH really dove in as one of the earliest NIH institutes to invest in this area. And so in the first few years, we funded our first exploratory studies using iPS cells to study a variety of autism spectrum disorders, schizophrenia, mood and anxiety disorders, and other disorders within our institute's mandate. In 2010 to 2011, we decided to stand up capabilities at the NIMH repository to allow production, banking, and distribution of iPS cells and their source cells. And so this was integrated with the NIMH Genetics Initiative to facilitate the availability of these resources in an integrative way, both for functional genomics and for cell-based assays. There's currently 278 iPS cell lines currently available. That's a little bit misleading because we have an estimated 700 to 800 more donor lines in process or expected from active projects. So we expect that this will increase fairly substantially over the next year. I'll note that the NIMH sharing policy was first promulgated in 2012 and has since been updated in 2021. And this states an expectation that NIMH grantees will submit their source cells and their reprogrammed cells to the NIMH repository. And included in that is a stipulation that subject consents must allow for de-identified data and resource sharing to both academic and industry investigators, including use for genetic studies, and also includes special considerations regarding the generation of iPS cell lines, both in terms of quality control metrics and the provider of the derivation services. I've included a link here to what the policy states. There have been some unique challenges to the NIMH iPS cell banking effort, which is a product of how we do business. We are a granting organization. And so a decision was made early on that with rare exceptions, NIMH funds iPS cell projects that are focused on biological discovery and quote unquote disease modeling. And we do this through research grants, individual research grants that are proposed through applications by individual investigators. And we do this rather than a concentrated, focused resource generation effort that you might see with other organizations like the California Institute of Regenerative Medicine. For that reason, the timelines for generating usable lines and data are longer than they would be for genetic studies and for these resource generation efforts. And this is because we generally allow an embargo period for the duration of the project period before the lines become available for distribution. Also, the consenting process changed over time. Early on, projects were not, the consenting process did not encourage consents that comported with centralized banking and distribution, but this is still changed. And we, this was an issue for a small number of lines early on, but pretty much now lines that go into the repository are consented for general research use and broad distribution. Also, the production methods are a little bit different for iPS cells than they are for LCL production or DNA directly from blood samples. And this is because we allow iPS cell line production to be done externally by individual academic core facilities rather than requiring all to be done at the NIMH repository. Since then, with our latest policy, we've really pushed forward to having lines centrally derived by the NIMH repository. And in fact, this is the case with the Phelan-McDermid collection, where all of the biomaterials from the recruitment onto the generation of the iPS cell lines was done by the repository itself. There are some unique opportunities with the NIMH collection. So first of all, it is an integrated collection with our genomics research efforts so that all materials can be processed and stored in the same facility, which makes it easier for both submitters and requesters. And NIMH is among the very few NIH institutes or centers that coordinates the collection of biospecimens and data for both genomic and functional studies at a large scale. As an example of this, the NIMH repository currently houses approximately 400,000 samples, either as cryopreserved lymphocytes or lymphoblastoid cell lines. The obvious implication is that these are a potential source for the expansion of iPS cell resources, and some of this has already been done with archived samples. A caveat with this is that cryopreserved lymphocytes are not what we would consider a catalog item in the sense that you can place an order. What you would do is you would negotiate with NIMH program on potentially accessing these samples, but the stipulation would be that the NIMH repository itself would take the samples from its own collection, reprogram them into iPS cell lines, and you would get a sample of those once they're derived. The remainder would be banked and be available for broad distribution. This is just to show you the production capabilities of the stem cell facility within NRGR. The main pipeline involves Sendai, cytotune 2, although episomal reprogramming is an option. They reprogram from a number of different source cells, primarily through blood, but they'll also do reprogramming from fibroblasts, urine, or other sources. They do have a standard quality control panel that they use, but there's additional quality control add-ons that you can include, and these are done as a menu item that you can choose from. And they'll also do gene editing as well as biobanking of both the iPS cell lines as well as the various source cells that are provided. So I just wanted to give you an idea of what the website looks like and how you would go about exploring what's available at the NIMH repository. So this is a screenshot of the landing page at the NIMH repository. And if you look at the upper left-hand corner, one way that you can explore is to look at the different studies that are available. We currently have over 200 studies that are listed in the NIMH repositories, and the studies usually correspond to individual NIMH grants. So it will list the grants and they will list the sites that are associated with that particular study. The sites can either be individual physical locations in which recruitment has been done, or sometimes in the case of iPS cell lines, it can refer to different categories derivation of samples that were originally collected through another site. I've highlighted here study 115 by Ricardo Dolmetsch and Joachim Hallmeyer. This is the study originally associated with an NIMH grant, but it included supplemental funding, which supported the recruitment of subjects from the Phelan-McDermid Family Foundation at an annual meeting that I'll describe in a few moments. One thing I'll note is that different studies are classified and released under one or more different distributions. And these will be listed, for example, as iPSC distribution 1, 2, 3. I think they're currently at 11. And as more come online, they're added to create a new distribution. The Phelan-McDermid syndrome biospecimens are listed under the iPSC distribution, but they're also listed under a separate Phelan-McDermid distribution. And when you go into the website, you'll be able to access the different distributions based on category. You can also look at the stem cell collection. You can either, again, look under the Explore Our Data tab, or there's a specific button that allows you to access it directly. And what this will show you is it allows you to filter based on pedigree, biosample, or genetic diagnosis. And this gives you an example of what will show up. One thing I will notice, and this is a caveat, we expected the Phelan-McDermid Syndrome Foundation collection to be available already. There's been a bit of a backlog in the curation process, so it's not available as of today, but we expect it to be released any day now. And I'll be sure to let everybody know. At a minimum, I'll send this through Geraldine, and she can pass it on. But we're I've been told that it is next in line to be released, so we're keeping our fingers crossed that it'll be soon. One thing I'll note is that the ASD, both ASD and genetic variants with a high incidence of ASD don't currently make up a lot. This is 21 total here. We expect this to increase pretty dramatically over the next 6 months or so, because we have a number of largish studies that are getting ready for distribution, which should probably get us into the I would say 60 to 80, actually probably closer to 100 over the next 6 months or so. One thing I will note is that this is a controlled access collection. So when you go to the website, you will be able to get high-level aggregate data, but what you won't be able to get is individual data that gives you information as to the de-identified information related to individual subjects. There is a process for requesting access that I'll go into in a moment. So let me talk now about the Phelan-McDermid syndrome collection. So as Geraldine mentioned, this was collected in the summer of 2012 at a Phelan-McDermid Syndrome Foundation Family Conference in Orlando. And we worked very closely with the foundation to put them in contact with an investigator team led by Joachim Hallmeyer, Jonathan Bernstein, and Ricardo Dolmetsch, who was at the time at Stanford University. And they oversaw the recruitment and the biospecimen collection. This was a great example of a really smoothly working partnership between a number of different parties, including the Family Foundation, NIMH, and these investigators. In the end, they recruited 134 subjects, which were kids and at least one parent, consisting of 55 pedigrees. All of these individuals donated biospecimens in the form of blood. Some were able to donate skin punches for fibroblasts, and the blood samples were processed and stored as cryopreserved lymphocytes at the NIMH repository. At the time, we didn't have funds available to do reprogramming into iPS cell lines, so they stayed that way for quite some time. But fairly recently, Ilya Singech, who was at the time led the NCAT stem cell translation laboratory, commissioned the 34 of the subject samples into iPS cell lines. These will be very shortly available for broad distribution, including to industry. This consists of 4 trios in which both the child, the mom, and the dad are represented in terms of iPS cell lines. We also have 4 duos where the child and one parent is represented. And then we have an additional 14 iPS cell lines where there is only the child represented. So that's 34 donor iPS cell lines in total. These, again, are integrated into the NRGR study 115 as site 726. So when you go into the website, you'll see where that's located. So how do you get access to these? When you first go into the website, very prominently, there will be an arrow in the lower left-hand corner for requesting access. And when you click that button, you'll come up with this instructions that shows up on the right. Now, what do those instructions entail? Well, first of all, what you need to do is you need to create an account using your organizational email address. Then what you do is you prepare a request package that you, once you generate the documents, you compress that into a zip file. What this is going to contain is a cover letter, which includes some general information, but also stipulates that if your research project isn't supported through NIMH, that you are prepared to pay all access fees. And the access fees are somewhat different between academic requesters and industry requesters. You'll also include a biosketch, a document that states the resources and environment that are available for you to do the research. And generally, this doesn't need to be more than a paragraph or 2. Funding information. For an academic research, this would be, federal or private foundation funding. For an industry investigator, this would just simply be a stipulation that your company is paying for the work. A concise description of the study that you're proposing to do, as well as a signed distribution agreement. And the distribution agreement is on the website, and you would simply pull that down and have that executed. And then additionally, if you're obtaining lines, you would fill out a data resource sharing plan if you're intending to generate genomic data from the lines, or if you're planning on generating genetically modified derivatives. The repository has the option of requesting that these be shared back into the facility. And then you would submit this into the portal using this request access form, which again is linked from that page. Now, the request is reviewed by a data access committee that consists of NIH program staff. And this is based on the experience and qualifications of the applicant PI and the co-investigators, ethical considerations of the use, the PI's familiarity with the characteristics, limitations, and strengths of the data set or the biospecimens, the different aspects of the research design and the research environment, and whether the PI has the resources to support the study, as well as whether they've adequately addressed sharing of either the data or any resources. And once that's done, the decision will be conveyed by email, and then you'll be given access to both the data and the ability to request biospecimens. I do suggest that whenever you make requests, that you try to be generally inclusive of what it is that you might need, because when new collections come online, you'll have access to everything up to the collection, the most updated version of the collection that you need. But if you want to update or make more timely your access based on what has been generated most recently, you would need to re-request access. And so for that reason, if you're specifically interested in accessing this Phelan-McDermid collection, I would suggest that you wait to put in the request until it's known that the Phelan-McDermid syndrome collection is available for distribution. And I will certainly let people know when that happens. That's it in a nutshell. I kind of ran through things fairly quickly, but I hope that gives you a good idea of what's in the collection and how to access the Phelan-McDermid samples as well. Thank you very much.
Thank you, David. That is super helpful. I want to give people an opportunity to ask questions if they have any. I guess I will lead with one, which is there anything different about the access request or the review for companies versus academic investigators?
No. I mean, they're all assessed based on the proposed use, the availability of the resources and wherewithal to actually do the research, and all of this is sort of need to know documentation. What is the reason why you need to have access to these samples? But generally, if people have the documentation in place that shows that there is a reasonable case for them having access to the materials, we are fairly open to sharing these.
I guess another question that people are probably wondering about is, is there a cost for industry? And if so, where can people find that information?
The cost is on the website, and unfortunately, that is something I meant to look at before I came on the talk. But it's significantly more for industry, but it's not an outrageous cost. But it's been updated recently, and I forgot to look at what it was.
Okay. Do you mind just sending me a link to that, and then I can distribute that?
I sure will.
Especially when you confirm that the samples are available, and I'll send out an email to everybody about that.
Right.
Yeah.
And I've just allowed people to unmute themselves for now. Do we have time for one more question, David, before you have to go? I know you have a hard stop.
Yeah, sure. If anybody has one more question, I can answer.
Okay. All right. And if anyone has any further questions after the webinar, then you can always send it to Geraldine or myself. So I'll go ahead and put everyone back on mute.
Great. Well, thank you very much, everybody. I'm glad I was able to present this, and thanks very much for the invitation.
Thank you so much. We really appreciate it.
Bye-bye.
Have a great day.
You too.
Okay, well, I'm really delighted to introduce you to our next speaker, Dr. Alex Shcheglovitov, Assistant Professor of Neurobiology at the University of Utah. He also holds appointments in psychiatry and biomedical engineering. I met Dr. Shcheglovitov probably, I don't know, 12, maybe even 12 years ago, or maybe longer, when he was a postdoctoral fellow in the lab of Ricardo Dolmetsch at Stanford, where he was very literally pioneering the use of iPSCs to study brain development and Phelan-McDermid syndrome. And I think his work was the very first time anyone really tried to use this approach to study an autism-associated condition. Since then, Dr. Shcheglovitov has continued to do groundbreaking work characterizing cortical neurons and now brain organoids. So, Alex, we're so happy to have you, and I'll let you take it away now.
Okay, thank you so much, Geraldine. It's really great to be here, and it was nice to hear what's available at NIH for research on Phelan-McDermid syndrome. And yeah, I'm going to start my presentation immediately. So, let's see. Okay. Do you see my slides? Okay, great. Yeah, again, it's nice to be here. I'm glad that you have this meeting to kind of to bring together people from industry, so think creatively about how stem cells can be used to develop new therapies for patients with Phelan-McDermid syndrome. And as Geraldine already mentioned, I was a postdoctoral fellow at Stanford University in the NIMH lab, and now I have my own lab in the Department of Neurobiology at the University of Utah. And yeah, the work that I will present today, and I have only 10 slides, have been funded over the years by NIMH, and David described the resources available. And so part of the funding that he also described was related to this work, and also current projects in my lab are funded by NINDS, and in the past we had some funding from other foundations. And I would like to also to mention that importantly, when I was a postdoctoral fellow at Stanford, my work was co-funded by Phelan-McDermid Syndrome Foundation and Autism Science Foundation. And it was really important support that came at that time. So in my lab, we study human neurodevelopmental disorders associated with autism, intellectual disability, and epilepsy, and we use 3 different model systems. And here you have a summary of those models. On the left side, these are the neurons, stem cell derived neurons, iPSC derived neurons, labeled with different colors. We can culture them in 2D on a dish, and we can use fluorescent proteins to label them with RFP, red fluorescent protein, or GFP, green fluorescent protein. And it's really nice system to go after cellular molecular phenotypes, because cells are accessible and we can easily manipulate the cells. We can study their functional properties and look at the synapses, composition of synapses, and we publish the description of the system in our Nature 2013 paper. So more recently, we started also engrafting human neurons into the mouse brain. And this is a great system to study network-related questions, because as we all know, cells that grow in a dish, they are different than cells that grow in the brain. And so here we try to recapitulate brain-like environment by transplanting human neurons into the mouse brain and then growing them for a number of months, and then looking at the properties of human neurons in the mouse brain using electrophysiology, and also rabies virus tracing, which allows to to map connections between human neurons and mouse neurons in different brain regions. And the final model system that we recently published on is human brain organoids, and we developed a new method to generate telencephalic organoids from induced pluripotent stem cell derived neural rosettes. It's out. It was published in Nature Communications in 2022. So if you would like to learn more details on this system, I encourage you to look up this paper. Well, again, as you all probably know, that the great advantage of induced pluripotent stem cells is that they have the same mutations and the same genetic backgrounds as the patient cells. So when we take blood samples or skin samples, in this case we have skin samples, and we use the skin cells to generate stem cells, we know that the stem cells, they, number one, have the same genetic background and the same mutations as patient cells, and number two is that the stem cells are pluripotent, so they are flexible and they can be differentiated into any cell type that we want to study. So for example, if we want to study brain defects, we can differentiate the cells into neurons or other brain cells. Or if we're interested in studying like cardiac problems, other problems, the cells can be differentiated into cardiomyocytes or any cell types of interest. So in my lab, we differentiate them into neurons. And so this is a summary of our work from 2013. And so here we used induced pluripotent stem cells that were obtained from 2 individuals with Phelan-McDermid syndrome and 2 control pluripotent stem cell lines. And so we generated stem cell derived neurons, and on the top image, we have a control neuron labeled with green fluorescent protein and patient neuron labeled with red fluorescent protein. So we co-culture them together to reduce cultural artifacts that are associated potentially with different number of cells if we culture them separately. And here we studied the functional properties and synaptic properties of these co-cultured neurons. We recorded synaptic both excitatory and inhibitory synaptic transmission, and it's sort of functional assay that allows to assess how these neurons communicate with other cells in a dish. And as you can probably appreciate from the recordings, is that neurons that were derived from patients, they showed significantly reduced number of these spikes. And those are called excitatory, spontaneous excitatory postsynaptic currents, which is indicative of impaired excitatory synaptic transmission. We then performed immunostaining with synaptic markers, and this is bottom panel of the slide, with synapsin-1, which is presynaptic marker, and Homer-1, which is postsynaptic marker, and we compared the number of excitatory synapses on patient and control neurons that were co-cultured together, and we found that patient neurons showed significantly reduced number of excitatory synapses as compared to control neurons. So this result suggested that neurons that were derived from patients with Phelan-McDermid syndrome, they have impaired excitatory synaptic transmission. So we also, what we did, we were able to rescue AMPA-mediated excitatory synaptic deficits in these neurons by delivering exogenous SHANK3. So we generated lentivirus and we infected patient cells with lentivirus carrying exogenous SHANK3, and we showed that this rescues AMPA-mediated excitatory synaptic deficits, but not NMDA-mediated excitatory synaptic deficits. We also showed that that treatment with Ig insulin growth factor 1 also compensated excitatory synaptic deficits. So it increases excitatory synapses, but it worked through a different pathway. So it didn't do, it's actually reduced the expression level of SHANK3, but it increased the expression level of PSD95. And it was an interesting and surprising observation. So just switch gears here, and here more recent work that has been done in my lab. So we use CRISPR-Cas approach to generate SHANK3 deficient stem cell lines. So, and the idea was that when we study patient iPSC derived neurons, the cells have SHANK3 deletions, but also some other genes that are deleted in that region. And so since we were not able to rescue all the deficits with lentiviral SHANK3, we thought that we need to have isogenic lines that only have SHANK3 deletion and all other genes in that region intact. So we generated two different SHANK3 engineered lines. One line has homozygous deletion of SHANK3 incorporating the tail part of SHANK3. And another line is hemizygous, containing hemizygous SHANK3 deletions that has complete SHANK3 deletion from one chromosome. And so we then use this control engineered stem cells to study the properties of SHANK3 deficient neurons upon transplantation in the mouse brain. And it's the cartoon is depicted on the bottom panel of the slide. So you can see that we can differentiate isogenic control stem cells and SHANK3 deficient stem cells into stem cell derived neurons. Then we can label them again with different fluorescent proteins, GFP and RFP, and then we can transplant them into the mouse brain, into the same region, and wait for a number of months, and then we can cut slices and study the properties of co-transplanted control and SHANK3 deficient neurons in the same brain. So here you can see. Again, this is a heavy slide, but so this the results of this research suggest that SHANK3 deficient neurons, when transplanted in this mouse brain, they have also impaired excitatory synaptic transmission, and the deficits were related to the amplitude and not to the frequency of spontaneous synaptic events. And then when we used this stimulation to induce synaptic responses and record both AMPA and MDA responses, we observed that SHANK3 deficient neurons showed impaired AMPA, but not an MDA-mediated synaptic transmission, which has resulted in reduced AMPA to an MDA ratio. We also, so this is one time point that we investigated in the study. We investigated later time point when we waited for 1 year or more than 1 year to record these transplanted neurons in the adult brain. And what was interesting is that we found different synaptic deficits. So in older animals, we didn't see the deficits in amplitude, but we observed the deficits in frequency, suggesting that SHANK3 may result in different functional deficits at different developmental time points. And this could be potentially related to the observations that people did previously studying SHANK3 deficient mice. So maybe depending on the time point that you use for your experiments, you may see different functional synaptic deficits. Yeah, in the study, we also observed morphological deficits and reduced number of spines on SHANK3 deficient neurons as compared to control neurons. Finally, more recently, as I already said, we developed a new method for studying human telencephalic development using organoids. And so what was different from other approaches that people use in the field is that instead of starting from induced pluripotent stem cells, we started from isolated individual neural rosettes, and then we grow this isolated rosettes in suspension for a number of months, and we can see that this rosettes result into organoids, and organoids have predictable composition and organization of cells inside. And so we did really thorough characterization of different cell types that are producing these organoids at different developmental stages. I don't have the slides related to this, but if you're interested, I encourage you to look this information in our recent paper. So now, related to SHANK3, we use this approach to generate organoids from isogenic control and SHANK3 deficient stem cell lines. And for these experiments, we used homozygous, the line with homozygous SHANK3 deletion. So first we'll look at the composition of cells, and it was important because no one looked at whether SHANK3 affects the early stages of brain development. And if it affects, then it doesn't even make sense to look at the synaptic transmission, because maybe the deficits are related to this early stages of cell specification or cell proliferation. So we did single cell RNA sequencing, and it's an unbiased way to look at the cellular composition. And so you can see on top, you see single cell RNA sequencing results obtained from isogenic control organoids. At the bottom, you see results obtained from SHANK3 deficient organoids. And what we found is that the composition of cells in these organoids was relatively similar. So we probed 2 organoids from isogenic control lines and 2 organoids from SHANK3 deficient lines. And here you see the plot kind of showing the composition of cells, and you can see there were no major differences in the cell types that we found in these organoids. So this suggested to us that SHANK3 probably is not the gene that affect this early stages of brain development. And here we also, you can see like heat map for the expression of different cell type specific markers. Like for example, RB-FOX3 is a marker of mature neurons. You see that it's relatively similar proportions. FASF is a marker of deep layer cortical neurons, also similar proportion of those neurons. SETB2 is a marker of superficial layer cortical neurons, as you can see, similar proportion of the cells, and so on. So we see relatively similar composition of cells in control and SHANK3 deficient organoids with no major differences. So then we decided to look again at the functional properties and synaptic properties of neurons in these organoids. For synaptic properties, we did immunostaining with synapsin-1 and SHANK3. We observed that SHANK3 deficient organoids showed reduced number of synaptic puncta which is again consistent with the idea that SHANK3 deficient neurons have impaired synaptic properties, excitatory synaptic properties. We also look at the intrinsic excitability, and this is sort of ability of neurons to fire electrical pulses. And here we compare different intrinsic properties of controlling SHANK3 deficient neurons for both hemizygous and homozygous SHANK3 deficient organoids. And as you can see that we found that SHANK3 deficient neurons have impaired intrinsic excitability, which is consistent with not impaired, but dysregulated intrinsic excitability, which is consistent with overexcitability of the cells as compared to isogenic controls. And again, this result is consistent with the result of our previous study on cultured 2D neurons, and also this result is consistent with the results of another study by Tom Sudhoff that also showed that the SHANK3 deficient neurons have impaired intrinsic excitability. So finally, we wanted to ask a question. Okay, we have these deficits that are related to functional properties, but what are the mechanisms? What can we learn from this system? And so for this, we decided again to take this blue sky approach, right? Unbiased approach to look at the differentially expressed genes. And we did bulk RNA sequencing on hemizygous SHANK3 deficient organoids and isogenic control organoids. And here you can see volcano plot that shows differentially expressed genes. On the left-hand side, the genes that are downregulated in SHANK3 deficient organoids, and on the right-hand side in red are genes that are upregulated in SHANK3 deficient organoids. And as you can probably appreciate, that most of the genes were downregulated in SHANK3 deficient organoids. When we did Panther cell pathway analysis to see if those genes kind of belong to a particular pathway, we found that the cadherin signaling pathway is upregulated in the downregulated part of this plot, suggesting that SHANK3 deficiency may result in disrupted cadherin signaling in the cells. So to confirm this result, we decided to do it again, bulk RNA sequencing, but on another samples that were generated from a patient line, from a patient that have only partial SHANK3 deletion and some other genes in the tail of the chromosome that are believed to be not that important for the neuronal deficits in patients. And so again, so here we have 6 organoids containing 3 engineered organoids and 3 organoids that were generated from a patient with Phelan-McDermid syndrome, and we have 6 organoids from 3 from isogenic control lines and 3 from unrelated controls. And again, as you can appreciate, on the left-hand side, we have impaired cadherin signaling, and it was significantly enriched. So again, supporting the idea that SHANK3 deficiency lead to the disrupted cadherin signaling in SHANK3 deficient organoids. So now to summarize this result, so what we know that SHANK3 deficiency results in synaptic and intrinsic and neuronal deficits, but we don't understand the signaling cascades. And so this result suggests that this signaling cascade could be related to disrupted cadherin signaling. However, we don't understand how SHANK3 deficiency disrupts this cadherin signaling and whether restoring this cadherin signaling could restore synaptic deficits. And that's something we're currently working in my lab to understand the links between SHANK3 and clustered protocadherins and clustered protocadherins and disrupted synapses. So to summarize what I just told you, we detected impaired excitatory synaptic transmission in SHANK3 deficient human neurons, and that was published in Nature 2013. We observed reduced dendrites and dendritic spines in SHANK3 deficient human neurons that were transplanted in the mouse brain, and that was described in Molecular Psychiatry 2021. Then we generated a new method for making telencephalic organoids from neural rosettes. and we found impaired excitatory synapses and elevated intrinsic excitability in SHANK3 deficient organoids, and also we observed impaired expression of cluster 8 protocoherence in SHANK3 deficient organoids, and these results were reported in Nature Communication 2021. So with this, I would like to acknowledge people who did all this work. These are the people in my lab at on different years. You know, we like to work hard and we like to have fun together. And yeah, the funding support that comes for this research from different foundations. So yeah, with this, I'd like to stop and happy to take any questions.
Okay, I've just allowed folks to unmute themselves if there are any questions.
I'll jump in. This is Paulina. Good to see you, Alex. Thanks very much for this talk. I'm just curious if you have used any of the collection of organoids that you've described for any kind of drug screening activities, and if so, like what type of screens you looked at?
Hey, Paulina, we haven't used them for drug screen activity, and one of the reasons is that as I showed in my presentation, the deficits we observed in SHANK3 deficient organoids were related to the later stages of neurodevelopment, right? So the deficits we observed were functional deficits, intrinsic excitability deficits, and also excitatory synaptic deficits. And to study those deficits, you need to wait for like 5 months. Those are 5-month-old organoids, and you can imagine that it's a bit difficult to grow them for that long and then have a large number of organoids so they can be used for drug screening. So what we are currently trying to do is that we're trying to develop a drug screening on 2D neurons, so that we don't have to wait for 5 months, as we can wait for 3 weeks or 4 weeks, and then we can have some insights into the chemicals that can increase the expression level of SHANK3. And I didn't have the slide in my presentation, but in my lab we also generated an engineered line that have endogenous SHANK3 tagged with halo tag, and it's sort of halo is a kind of, it's a GFP-like tag that allows to visualize the expression of endogenous proteins, and also that we could use it for pull-down assays and kind of identify SHANK3 interacting proteins. So, and also it allows us to visualize the expression of SHANK3 in live cells. And so the idea is that we would like to generate neurons from halo tag SHANK3 expressing stem cell line, and then look at the SHANK3 expression in live cells, and then do treatments and see if we can find something that can increase the expression level of SHANK3. So this way we would target not the deficits we see in the cells, but we would target the SHANK3, so the expression level of SHANK3. Yeah.
That's really very interesting. It's really insightful. It's interesting. It's almost because the organoids are so complex to grow and so expensive to grow, I guess, to the 5-month stage. I guess it's almost like one has to view the complete 3D organoid as sort of a test system that would be similar to, like, if you had, non-human primate, like you had a marmoset or something, and you would ultimately treat it with, a drug and see what like systematic effects they were, right? You almost would sort of treat a brain organoid in the same manner, right? You wouldn't use it for, pre-screening of potential compounds. You'd use it sort of as a final sort of maybe system that you look at.
Yeah, yeah, that's exactly right. And we kind of we thought hard about this approach because the ultimate goal is to find some therapies, potential therapies for patients, and we published this review paper in Developmental Dynamics a few years ago, and in that paper we proposed that it's probably good to use organoids for getting insights into the deficits, because we can use bulk RNA sequencing, we can use single cell RNA sequencing to kind of to look for the global deficits. But then for screening, I mean, if we, depending on what kind of deficits we see in the organoids, we can set different screens. So for example, if we see functional neuronal deficits in organoids, we can set screens on neurons, and we don't have to use organoids for screens because it's an expensive and it's a really long process to make those organoids. But then, like, once we have the screen on neurons, we can go back again to organoids and confirm that, for example, the drug, a drug that we find in cultured 2D cells can also rescue the deficits in organoids. So it can be a kind of used as an initial platform to find the deficits, and as the final step is kind of in testing the drug. But I think in the middle should be something else that would allow to have a quick and quick high throughput screen for drugs.
Thank you so much. Thank you. Thank you for answering the questions, and thank you for your time today and your presentation. We really do appreciate it. We have just a few more minutes left, and we'd like to share some information about what we are doing in various projects, and we are going to focus on the Biomarkers and Outcome Measures Consortium, the pre-competitive consortium that we are building. And I want to share my screen with you all and go to the Biomarkers and Outcome Measures Consortium. What we would like to do with this particular consortium is to create a pre-competitive space where we can work together to facilitate a cooperative and globally aligned effort to focus on the identification and development of novel approaches to enable successful clinical trials. And so in the simplest of terms, we want to bring together interested companies to work collaboratively on the development of outcome measures and biomarkers. And this is the thinking behind the consortium. When CureSHANK was looking at building a consortium, CureSHANK engaged with many companies over the last years, and the conversations have been very similar, and they've converged on outcome measures and biomarkers. And clinical trials in disorders of the brain are hard. They're costly and don't always end in success. But your success is our success. And by working together, we can get there faster, cheaper, and more efficiently. Oh, excuse me just for 1 moment. I'm really sorry. Thank you. The consortium, if you take a look at this slide, the consortium is convened and overseen by CureSHANK, and we have an executive committee of advisors that advise the executive committee, and the executive committee is responsible for the mission, the vision, the strategy, and the governance of the consortium. And so if you look at who can participate in the consortium, companies who are interested in scientific areas with particular promise for the development of outcome measures and biomarkers for Phelan-McDermid syndrome are invited to participate. And so the companies that will participate as members can serve on the steering committee and project teams. And so why join the consortium? The consortium brings stakeholders to the table, and we bring solutions to the surface faster, and we want to make it easier to scale them by working together. We would like to pool resources to move the field forward as a whole. And it's very important that we can reduce duplicative efforts so that we can reach the same goal together. We want to tackle shared obstacles and create universal tools and create frameworks that we can use widely. And it's very important that we streamline regulatory approval of new outcome measures and biomarkers so that we're all speaking the same language when we go for regulatory approval for the therapies that we are looking to do together for the field as a whole. And above all, we want to make meaningful change. We want to drive change through a pre-competitive effort of working together. And we want to build community within the space for us to be able to advance our efforts together. And so we invite partners to consider to join the consortium. And the process is that you can contact me, and we will create a CDA and review a term sheet for participation, and then establish an agreement to join the consortium. And we understand that a decision to participate in the consortium takes some discussion with your company. We'd love to talk with you about any shared interest on particular outcome measures and biomarkers, and we're receptive to ideas because we want to build this community and work together. And I'll turn it over to Geraldine to close us.
Well, we're so glad that we were able to organize this really informative workshop today. Hope it was helpful to all of you. We really appreciate both Dr. David Panchision and Dr. Alex Shcheglovitov for joining us today and sharing. And if you have any questions, you're welcome to reach out to us. There's an email address listed right there, our website. And I think most of you know how to reach me. Feel just free to reach out anytime. And we hope to do another one of these again in the future. And if you have suggestions for topics, please let us know. We'll be happy to help connect you with other investigators and other resources in our field. Thank you again for joining us, and have a great weekend.